This is ameloblast. These are the short columnar cells with large oval nuclei. These are the parts of ameloblast in which the Golgi apparatus and centrioles are located in proximal part and mitochondria are evenly distributed. These are the parts of enamel organ. Coming to life cycle of ameloblast, in which the first stage is morphogenic stage. In this stage, the inner enamel epithelium is separated from the dental papilla by delicate basal lamina. Appearance of terminal bars. These are the cytoplasmic extension. Then mitochondria migrates to the basal region of cell. Then second stage is organizing stage, in which inner enamel epithelium sends signal to adjacent mesenchyme, which is dental papilla. Due to this epithelial and mesenchymal interaction, the peripheral cells of dental papilla differentiate into odontoblast. These are the odontoblast. Migration of centrioles and Golgi bodies to distal end of the cell, and nuclei extends to the proximal part of cell. This process is called as reversal of polarity. Due to this, the odontoblast lays down the predentin. When predentin is formed, it cuts off the nutritional supply from dental papilla to ameloblast. To supply nutrition to the ameloblast, there is a reduction in volume of stellate reticulum, and the distance between the capillaries and stratum intermedium are shortened. After this, the ameloblast receives nutritional supply from capillaries, which is supplied from the outer enamel epithelium. This process is called as reversal of nutritional source. Coming to the third stage, which is formative stage, in which there is numerous secretory vesicles are seen in ameloblast. Then development of blunt cell process on the ameloblast that even penetrate the basal lamina and predentin. These blunt cell process are called as Sturm's process. Enamel matrix formation begins over the first layer of dentin, which secretes enamel protein. These are amelogenin and non-amelogenin. Then appearance of Sturm's process. These are the conical, blunt or pyramidal extension of ameloblast cytoplasm that protrudes into the developing enamel matrix. Then there is distal terminal bars which separate Sturm process from cell proper. Coming to maturative stage, in which enamel maturation occurs when most of the thickness of enamel matrix formed in incisal or occlusal area. Ameloblasts are slightly reduced in length and are closely attached to the enamel matrix. Then the cells of stratum intermedium lose their cuboidal shape and assume a spindle shape. Ameloblasts display microvilli at the distal extremities. In this stage, the cytoplasmic vacuoles, which contains enamel matrix protein, are present. The function of cytoplasmic vacuoles is absorption. The cells of stratum intermedium and outer enamel epithelium can no longer be differentiated into ameloblast, and these cells form a stratified epithelial covering of enamel, which is known as reduced enamel epithelium. The function of reduced enamel epithelium is to protect mature enamel from connective tissue by separating it from tooth until it erupts. Enamel organ retract from cervical edge of enamel, and then the adjacent mesenchymal cells may deposit a fibrillar cementum. Then coming to last stage, which is Desmolytic stage, in which reduced enamel epithelium proliferates and induce atrophy of connective tissue by fusion of reduced enamel epithelium and oral epithelium. After its fusion, it helps in eruption of teeth.